Hello everyone. In this lecture, I will be covering cell cycle, mitosis, and meiosis. Here's a fun fact that it takes about 18 to 20 hours for the cell to go through interphase and takes about 2 hours for mitosis or meiosis to happen. Now let's get into some details about cell cycle. Now it's only in biology that multiplication and division means the same. In cell cycle, the cells multiply to make two daughter cells, and the cells divide to make two daughter cells. The cell cycle is an ordered sequence of events that extends from the time a cell is first formed from a dividing parent cell until its own division into two daughter cells or four daughter cells. Now we're going to talk about why there are two different types of divisions. Mitosis occurs in your somatic cells. And why do we need mitosis? Because it's the process that helps in growth, embryonic development, repair, and rejuvenation. Meiosis, on the other hand, takes place only in the germ cells of the body. Those are the sex cells. And it's a reduction process. That means the cell starts out with a diploid number and it ends up with half the number, which is called the haploid. Now, we want to ensure that the cycle is completely accurate. So there are major checkpoints, and these checkpoints are very crucial because they ensure that all the processes are completed before the cell can move into the next step. Now, during G1 phase, the cell is committed to division, and it ensures that all the growth factors are present for the cell cycle to continue. In G2, the synthesis phase or the S phase has occurred before that. So the G2 phase, the cell ensures that all the chromosomes have been duplicated properly. And finally, during the M phase, each and every homologous chromosome should be attached to the spindle fiber. So, there are some factors that are very important, and these are external factors that influence the cell division. So in this case, we're going to talk about three external factors. Number one is growth factors. There are over 50 growth factors, and they are the proteins that are essential to stimulate the division. Now, density-dependent inhibition is the second factor. What does that mean? For example, think about it. You get a paper cut and your skin opens up. So those cells are now separated. Those cells on the edge of the cut, they will start dividing immediately. But as soon as they divide and patch up your skin, and one cell membrane touches the other, the cells will stop dividing. So that is the density-dependent inhibition. And these cells, when they're dividing, they have to be anchored to the surface. If they get dislodged from the surface, the cell division will stop. So this is an overview of mitosis. I would like you to refer to this slide once I explain everything, and you can come back and review it. The pictures are pretty good. And here's some front loading for you. I would like you to watch these videos on YouTube, and then I will get into the details of mitosis and meiosis. So as I said before, interphase is the longest phase of cell cycle, and it's divided into three different subphases called G1, G2, and S phase. The G1 phase, the cell grows and all the organelles duplicate. During S phase, all the DNA replicates and makes duplicate copies of itself. So if you have 46 chromosomes, as in human cells, now during S phase, they have doubled up and the total number of chromosomes are 92. And these identical copies are called sister chromatids. G2 phase is the final growth phase in which the cell is ready to move into mitosis. So, the first phase of mitosis is prophase. During prophase, there are four things that are going to happen. Number one, the nuclear membrane begins to disappear at the beginning of prophase, and by the end of prophase, it's completely gone. The centrioles divide into two, and they start moving to the opposite poles. And these centrioles start making spindle fibers. All the chromatin, the chromosomes which were indistinct during S phase of interphase, now condense and they become very distinct and visible. Now during metaphase, all the chromosomes line up on the equator of the cell, and spindle fibers are attached to the centromere of each of these chromosomes. During anaphase, the spindle fibers begin to retract, and that retraction pulls the sister chromatids apart. And as these sister chromatids move apart, each side of the cell is going to have all 46 chromosomes, 23 from mom, 23 from dad, on each side. During telophase, which is almost opposite of prophase in many ways, the nuclear membrane begins to reappear. The spindle fiber has completely retracted. The chromosomes they unwind and start becoming indistinct again. And there is a cytoplasmic furrow that starts to begin on the each end of the cell, and the cell is more elongated. During cytokinesis, the cytoplasmic furrow deepens, the cell splits into two, and that is the end of cytokinesis. In plant cells, instead of cytoplasmic furrow, there is a formation of cell plate that divides the cell into two daughter cells. Now let's move into meiosis. Before that, let's talk a little bit about a couple of vocab words that you should know. Now, when mitosis goes uncontrollable, it results into formation of tumors and cancer. There are tumor suppressor genes that ensure that all the mutated cells are removed, preventing any formation of cancer and tumors. Oncogenes are cancer-causing genes, whereas proto-oncogenes are the normal genes that become mutated. So, let's talk about meiosis. Meiosis is production or formation of gametes. This is basis of sexual reproduction, and it's a reduction process taking place only in the germ cells. Now, before we move into the phases of meiosis 1 and 2, let's understand the differences between homologous chromosomes and sister chromatids. The homologous chromosomes have the same genes, but they are not identical. That means they can be dominant or recessive, and the genes are from mom and from dad. So let's say this chromosome came from dad, this came from mom. They have same genes, but those genes can have different expressions. So that makes up a homologous chromosome. Now during S phase, this chromosome made a copy of itself, and this chromosome made a copy of itself. So these two chromosomes now, the ones that we assumed are from dad, are identical. These are the two sister chromatids. Likewise, these two are the sister chromatids. But these homologous chromosomes are the ones that have the genes from mom and dad. So here's an overview of meiosis and mitosis. Diploid organisms in their reproductive organs contain cells. Those cells are going to undergo meiosis to make haploid gametes. These gametes fuse together to make by the process of fertilization and undergo mitosis to make diploid organism. So how meiosis have half the number of chromosomes? So let's look at this. This is interphase. During interphase, these are the homologous chromosomes, and each of these chromosomes will make an identical copy of itself during the S phase of interphase. So these are the two sister chromatids, but the overall pair is a pair of homologous chromosomes. During meiosis 1, homologous chromosomes split. Then realize that there is no interphase after meiosis 1, and these homologous chromosomes or the sister chromatids will split during meiosis 2, reducing the number of chromosomes to half and making 4 daughter cells. And these 4 daughter cells can have different types of chromosomes, and we're going to talk about that diversity in the next slide. So one more time, homologous chromosomes separate during meiosis 1 and sister chromatids separate during meiosis 2. So let's talk about genetic diversity, which comes from 3 different steps. Number 1, half of the genetic material comes from each of the parent. So that brings in diversity by itself. The second phase where diversity is more enhanced is in prophase 1, where crossing over occurs. And lastly, the alignment of chromosomes in metaphase 1 at the equator, that's the random alignment, that brings in more genetic diversity. So one by one, let's look at these processes that makes chromosome combinations more diverse. Crossing over takes place during prophase 1. We have our pair of homologous chromosomes. Researchers are not sure why or when these random exchange of tetrads occur, or the arms are switched around. The length of the arms that are switched around is also varied. So, as a result, we have parental type of chromosomes present, and we have the 2 recombinant chromosomes, which now are much more diverse than the original 2 chromosomes. The second stage where diversity comes in is at metaphase 1 by the alignment of homologous chromosomes at the equator. If you look at this, these homologous chromosomes line up, and here there's a random alignment which is different from these 2, and as a result we have 4 different possible combinations of chromosomes after metaphase 1. And let's go through meiosis 1 quickly, which is basically the S phase. G1, S, and G2 is identical to interphase that I discussed before. During prophase 1, the same 4 processes occur. Homologous chromosomes will switch arms, resulting in crossing over. Centrioles divide, move to the opposite poles, and we have homologous chromosomes lining up at the center. Homologous chromosomes split, and during meiosis 2, which is absolutely identical to mitosis, we have prophase 2 with no interphase prior to that, resulting into metaphase where all the chromosomes line up at the center. Sister chromatids split, making 4 haploid